Hello, superb students of science. Here's a plant growth experiment. How does light affect the growth of plants? And last week, we started 4 fava bean plants. 2 of them were grown in the light and 2 were grown in the dark. And we also took 20 seeds. 10 of them were germinated in light and 10 of them were germinated in darkness. And here are the results from this experiment. It's 1 and 2. They're grown in with light. You can see dark green color. It's Sunday today. Plant number 2 has some wilting around the edges of the leaves and some black coloring. Also, plant 4 has the same kind of black coloring, but you'll notice that these are much more pale. And over here also, the plants are becoming pale and what we call leggy. They're stretching out. They're trying to find the light. Whereas these plants, you can hear Russell hammering in the background. Russell, can you please be quiet while I'm talking about the plants for just a minute? Thank you so much. And down here, look at all these. These are the ones that were in the dark. Now look at all these seeds that have germinated in the pot. See them down there? Now the ones that are in the light do not have that same volunteer seed sprouting. It's the same exact soil, so the seeds are present in both. I mean, there's one right there, one little one, but it's not quite the same. Anyway, there's the difference. Dark, light. We're on day 4. It's the final day of the plant growth experiment. I actually got a new light during this process, and this is a full spectrum LED light that I have to help start tomatoes and peppers and all the plants for our garden. Let me turn this off, maybe that'll make it easier to see what we're doing here. Okay, so here are some of the bean seeds that have been in the light. You'll see we've got germination. We can take those out and count them. And I can see some green color there on the growing tips. And the first leaves, which are called cotyledons. It's a nice sunny day outside. And we've got our 2 plants that were in the light. There they are right there. See, my hand is about that big. So these have grown rather large. That's plant number 1. And this is plant number 2. Okay. Now the other plants in our experiment have been in the dark, and they're down here in this shopping bag. So let's take this off and see what we've got going on in here. Wow, these look completely different. Here are the 2 that were in the dark. You'll notice there was another seeds in here that have actually sprouted. Look at all these little sprouts that have volunteered. This is another fava bean that was cut off actually before the experiment started. And just take a look at those. We'll do some more analysis in just a minute. What do you notice about the color? Let's bring them up next to each other and see if this helps. It's a little hard to tell. We'll bring them into another area and see what you can see. Alright, the next thing we'll look at are the seeds that have been in the dark. And there they are. I do see some green in there, but that green is actually on, I think that's part of the paper towel actually. That's not part of the seeds. So these are the ones that were in the dark. Notice there that the roots and the first cotyledons are white. There's no green color. So let's keep them here in the dark. And now let's compare that to our other ones that were in the light. Let's just do this real quick and you'll see some green color there. Alright, let's do some analysis. Okay, there we go. We started 10 in each. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Alright, what do you notice about these Kentucky Wonder bean seeds that have been in the dark for about a week? Okay, we're going to move those down here. We're going to compare these. That's jar number 2. We're going to compare those to the seeds that have been exposed to light. These are actually grabbed on to the paper towel with their roots. They're trying to grow into the paper towel. So let's count these. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So we have 100% of the seeds are still here. Nothing disappeared. And what do we notice? So here are the ones that were in the light. Come on, focus camera. And here are the ones that were in the dark. I'm going to back up so you can get a view. That's jar number 1 in the light, jar number 2 in the dark. Okay, the 10 seeds that were grown in light are 11.6 grams. One, 2, 3, 4, 5, 6. And the 10 that were grown in the dark are 11.1 grams. Plant 2, plant 3, and plant 4. Alright, so first we'll make some qualitative observations. So what do you notice about plant 1? What do you notice about plant 2? What do you notice about plant number 3? And plant number 4. Alright, to compare, here's plant 1 and plant 4 next to each other. So you can see the difference in the color. Here's plants 1 and 2 in my left hand, 3 and 4 in my right hand. Trying to be gentle. And next, let's just count the leaves since we did count them at the beginning. 1, 2, 3, 4, 5, 6, 7, 8. On plant 1, on plant 2, 1, 2, 3, 4, 5, 6, 7, 8. Alright, on plant 3, 1, 2, 3, 4, 5, 6. But 5 and 6 are pretty small. And then on plant 4, 1, 2, 3, 4, 5, 6, 7, 8 leaves on plant 4. Alright, next we're going to figure out how tall they are, and I'm going to use this ruler, and I'm going to show you something I don't know if you can see, but on this ruler, at the bottom of this ruler, there's a space right here between the edge of the ruler and the first line, and I've already measured that. That's a ½ centimeter. Okay, so I need to calculate for that. So I'm going to measure here, and this is 23.6. Number 1 is 23.6. Number 2 is, wait, I need to back up. I did the ½ centimeter wrong. Sorry about that. We need to add the ½ centimeter. Number 1 is 24.3. Number 2 is 25.8. Number 3 is 23.5. And this one here, number 4, is 27.6. Those are all centimeters. And another thing that we could measure, which we didn't measure at the beginning, is the internodal space. So we could measure the distance between this node and this node, and this node and this node, and this node and this node, and compare that to the space between these. And what we might notice if we did that is some elongation. So let's measure the difference between the length of, how about the second node to the third node. So from this leaf to this leaf. So on plant number 1, if I measure that length, I get 6.1 centimeters on plant number 2. If I measure that length, I get 7.0 centimeters. On plant number 3, if I measure that length, I get, wowzers, I get 8.9 centimeters. And on plant number 4, if I measure that length, I get 7.3 centimeters. Okay, and the last thing we look at is that in the plants in the dark, do you see the soil? There were some other seeds, I guess, in the soil, and the ones in the dark, those other seeds were able to sprout. I wonder how that affects the health of the fava bean. Notice that the ones in the light, there is a volunteer sprout here, but there's not as many as there are over here. All right, that's something I noticed. All right, now that we've got the data, we need to enter that into our data table and do some analysis. And that's our next step. What we need to think about is what is the most important information here that will support or reject our hypothesis. Our hypotheses, right? We have 2. And the big question is, how does light affect the growth of plants? And there's another objective, which is to demonstrate that plants need photosynthesis in order to be healthy, right? And so what did we observe anything? Are these plants healthier and better off than these plants? Okay, what would show that? You know, does measuring their height show that? Does the length between the leaves show that? How can we present that to someone who's reading our scientific paper? And the same with the seeds, right? Here are the ones that were in the dark. And here's the ones that were in the light. Okay, these first leaves are called cotyledons, and you'll see that they both have some cotyledons, right? But these cotyledons look different than these cotyledons. Which are healthier? And why? And how can you prove it? Here is something I have noticed, is that I separated the 3 most developed seedlings from the 10. So the 3 most developed in the dark, the 3 most developed that are in the light. And I just looked at them for a little bit, and I noticed that there's a center root, right? And that's this root here, kind of like the taproot. And then off to the side, you have lateral roots that come off to the side. And I noticed there's kind of a cutoff here at about 2 centimeters, where in the dark, if I was to measure these, the length of one of those lateral roots, that's about 1 centimeter right there. Now let's look over here at the plant that was grown in the light. And there's a lateral root right there. I'm going to put this ruler up next to it. And that's a 3 centimeter lateral root. So I could go right now, we could count the number of lateral roots greater than 2 centimeters, and that might help us answer our experimental question. So let's go ahead and count those. That were in the dark, and the 3 most developed bean seedlings that were in the dark weigh 3.7 grams. And the 3 most developed that were in the light weigh 3.9 grams.